Got it. All right, now we got it. So Michael Meyer, aquatic ecologist, limnologist in the house. You got your start in studying lakes in a long time. Initially, you were an intern at the California Tahoe Conservancy, and I guess you decided that Lake Tahoe was not a big enough lake, so they sent you to Lake Valcon, which then I guess led you to get really intrigued by that part of the world, and you majored in biology, Russian studies, and international studies at St. Louis University, which is a really cool mix of topics, I would imagine. You later went back to Russia, State University, Research Institute, where you studied shrimp in Mendenhall, and then you went to get your PhD in Washington State University, where you continue to study limnology even now, and discussing the social impacts of food webs. Michael's background is also broadly in data science, remote sensing, and modeling, and those are the things that I think have brought him here to Madison, where he's a USGS Mendenhall postdoctoral fellow, working on hydrology and questions of modeling and remote sensing. And if you saw the abstract, you saw that this is kind of a piece of really kind of reflecting on many of those aspects and how we put them together in terms of data synthesis. So I'm really excited to see what you're going to tell us. And with that, I'll turn it over to you. Thank you.
Great. Thank you so much for inviting me. It's really exciting to be here, and thank you for that great introduction. Yeah. Hi, Michael. My name is Michael. I use he/him pronouns, and I'm going to be here to talk about a few of the projects that I've had a chance to work on, and why I think that it's actually, given 2020 and 2021, it's kind of hard to find some optimism. So hopefully I can at least inspire some positivity with this talk. And on this talk, it's a little bit different. I would love to say that I thought of it all on my own, but it's very much inspired by a talk that actually Stephanie Hampton gave at Ecological Society for America in 2020. So this is just kind of my spin. And even though I am at the USGS now, I need to put a disclaimer, all of this work was performed before I started at USGS. So, top it says 2020, a year in review. So I know 2020 was like 17 years ago or something like that, and kind of all of a mix of what's happened. So I figured we could just walk through what's happened over the past 20 months or so. 2020 was really a banner year. It started with this global pandemic that totally upended life as we knew it. International travel was curtailed. We all had to go into isolation, and we all had to go and learn how to work from home and how to be a little bit more isolated with our lives. And then this led to a rather uncertain job market and economic situation for many folks, and especially folks like myself who were about to enter the job market at the start of the pandemic. It was a really nerve-wracking, awful times. And then finally, while not exclusive to 2020 by any means, there was some racism and systemic inequalities were going on in society that disproportionately impacted underrepresented groups. And the COVID-19 pandemic, in tandem with this uncertain employment market, job market, financial market, disproportionately impacted certain communities, and especially those with intersecting identities, such as Black trans women. So to take away from this, 2020 was really an awful year. It was really bad, just to be a person. And it was also really hard to be a graduate student, or at least involved in scientific research at any point. And I have here this news clipping that saying that US graduate student anxiety and depression went up 50% in 2020. So not only did, and that's just graduate students, right? So I couldn't find the tweet, but there was a new study coming out about the toll on faculty advising graduate students during 2020 and the exhaustion that came with that. So not only was it hard to be a person, but it was also hard to be involved in a lot of the sciences, at least to the extent that we were used to being engaged in the sciences. Now, I am not somebody that should be proposing any solutions to how we go forward with any of this, right? But I do want to at least posit what I think could be a path And that's, I'm gonna try to, can I hide this? Hide floating meaning controls. Great. Wow, you learned a new thing 20 months in. So data synthesis, I think, could be that path forward. And what I mean by this is that there's more publicly available environmental data now than ever before. And a lot of it is at our fingertips. But the thing is that working with these data and efficiently wrangling and manipulating them into an analytically friendly format is a skill set in and of itself. Oops, come on. And I love this figure from Shannon Ledoux's paper that basically talks about the three main V's of data science or data synthesis. And real briefly, what that entails is we think of the variety of the data. You can think of how many variables are in your data set, where climate data might have a lot of different variables, genomics data might have relatively fewer. That needs to be considered. Also the velocity at which those data come in, or the frequency at which we're getting new information. Climate data, you can imagine coming in every few minutes, hours. Genomics data, biotic interactions, relatively fewer. And then these bubbles are sized by the data volumes, or how big those file sizes are. Or even though genomics data might not be coming in very frequently and might not have many types, those file sizes are huge. Whereas like biotic interactions that might have a bunch of different variables, not that frequent of updates, but the file sizes are pretty small. And often we in the environmental sciences and environmental synthesis are ultimately interested in merging these different bubbles. But to do so, you not only need to have disciplinary knowledge of climate data and genomics data, but also how to actually operationalize those joins. Now that's a skill set, that's something that can be learned. What can't be learned is the data, right? We need the data. But luckily, as I said, we live in a time where places like Data One, COASI for hydrological data, EDI for all types of environmental data, monitoring trends in burn severity for US fire data, Dryad for ecological data, NASA and USGS for remote sensing, hydrologic, geologic data, and even community science programs such as iNaturalist have put all of these data at our fingertips, where we can begin joining these data sets. That's a skill set, right? The act of joining them is a skill, but we're ultimately able to address questions at scales that were previously not possible. And so my goals for today are to kind of share how I've incorporated some of these data harmonization techniques and analyses into my own research, but I think I would be doing a disservice if I only talk about what I did and I didn't talk about how other folks can engage in the science. So hopefully I can offer some resources for learning these techniques. And there's always a chance that I could do a really bad job of 1 and 2. So at the very least, my hope is to give you folks some positivity and maybe even ignite some creativity in your own research. And the first thing I want to talk about is a study that came out in 2019 in Environmental Science and Technology. It's an evidence synthesis of the pharmaceutical literature in the environment, pharmaceuticals in the environment, and benefited from the great help of 2 awesome co-authors. And basically the lead-in to this study is thinking about pharmaceuticals and personal care products in the environment, or PPCPs. And there's been a lot of work done on PPCPs in the environment, but one of the big reasons why there's been so much work done is that it's an interdisciplinary research effort that brings in folks from civil engineering and environmental sciences and ecology and ecotoxicology. And indeed, this is not only just a disciplinary research effort, but a global research effort. So what I have here are some assets that are divided up by 5-year intervals. Countries are colored by the number of studies published within that country for which the first author is associated with an institution in that country. So I know that's really complicated, but we'll just go with number of studies from a country. And what we see here is that the pace of PPCP research has grown rapidly over the past 30 years. There's about 7,000 studies on this global map. Now, about 80% of these studies have been done since 2010. And about 90% of these studies have been conducted since 2006. Now that's a pretty rapid increase in this literature across multiple disciplines, but it shows that there's a lot of work going into understanding PPCPs in the environment. But given that rapid increase, we started wondering, are we seeing the whole picture? Or is research emphasis, given this rapid increase, is research emphasis evenly distributed within this literature corpus? And so how do we track these patterns in the literature? Well, what we did was we called a literature corpus from the Web of Science, which is this literature database, and we used this kind of catch-all search formula and did some QA/QC procedures to be sure that we were getting the literature corpus we wanted. And then we exported those from Web of Science and imported them into the R statistical environment. And we iterate through all, I think it's about 8,000 abstracts for instances of 70 different focal PPCP compounds, 15 different sewage treatment techniques, and 12 different ecosystem types. And when you do that, when you flag those instances within the abstracts, we can look at how the research emphasis has been partitioned. And as these pie charts suggest, research emphasis has not been equally distributed. And I'll talk about that a little bit. So let's first look at the most PPCPs that have been studied. So here on the x-axis, we have publication year. And on the y-axis, we have proportion of abstracts. And dots are colored by the number of publications within that year. What we see here is that non-prescription drugs, so things like caffeine, acetaminophen, nicotine, have been well studied throughout our time series, as well as antibiotics. And that makes sense, because nicotine, caffeine has been around for a really long time. One of the main tracers that folks use for sewage, or one of them for sewage pollution. Antibiotics, we know that there's going to be a biological response. So it makes sense why this has been dominated the literature. Less well studied are things like hormones and prescription drugs. Now, prescription drugs kind of make sense because there's constantly new drugs entering the market. So maybe not a lot of emphasis on a given certain class of drugs or type of specific drugs over the years. But hormones is interesting because about 20% of the literature, 20 to 25% of the literature samples for hormones. But in actual environmental surveys at the continental scale, approximately 80% of environmental samples contain at least one hormone. So we see this imbalance between what is actually in the literature and what's actually in the environment, or what we think is in the environment. Let me see that. And ditto for fragrances and antioxidants, where fragrances appear in the literature about 0.5% of the time, yet about 20% of environmental samples contain at least one fragrance. Antioxidants is actually where they match, where no abstracts reference antioxidants, and very few, I'm not going to say no, but very few environmental samples tend to have antioxidants. So it's very rare. Now, when we look at most studied sewage treatment techniques, the most prevalent forms tend to be activated sludge and water treatment plants. But more broadly, the most represented sewage treatment techniques tend to be forms of centralized wastewater treatment. Just noticed an error on this figure, but I'll say it on the next. I reran these like last week, so they could be most up to date for you folks. The least well represented forms of sewage treatment tend to be forms like decentralized wastewater treatment. So things like septic tanks, sand filtration, and also constructed wetlands. Animox should not be there. And this is important, and I think this is important, because when we think about it, decentralized forms of wastewater treatment in the literature constitute about 2.5% to 4% of the literature. But more than half of the world's population is on some form of decentralized treatment. And that includes the global north. Within the US, about 25% of US homes are on some form of septic system. About 20% of Europe is on a septic system, ditto for Australia. So this is also like that mismatch, right? And septic tanks tend to be less well regulated than centralized forms of wastewater treatment. So that presents I'm not going to say a knowledge gap, but at least an opportunity for expanding these foundations of centralized systems to decentralized. And last, for ecosystem types. Rivers and streams, moving waters, tended to be most well represented within this literature corpus. And also soils, subsurface environments also tended to be pretty well represented within this literature corpus. But least well represented were more stationary waters, things like lakes, wetlands, ponds, lagoons, marine environments, so oceans and estuaries, and also forests for terrestrial systems. And I think this deviation is important to think about because lakes, wetlands, ponds, lagoons, they tend to have longer hydraulic residence times than relative to things like rivers or streams. And that can create a situation where the contaminants could accumulate and pool and potentially elicit stronger biological responses. Also, ditto for marine systems like oceans and estuaries. Remember, more than half the world's population lives within about, 100 kilometers of a coast. Yet marine studies make up about 2% of the PPCP literature. So getting back to the question, are we seeing the whole picture? I think the optimistic way of looking at it is that, yeah, the literature has laid foundation in certain areas, such as groundwater, centralized systems, rivers, antibiotics, but there's a whole lot out there that hasn't really been explored as well, but we can use these foundations laying in certain spheres and then apply them to less frequently well-studied systems. Okay, that's chapter 1. So I'm going to transition kind of entirely, but keeping with that theme of using data in a different way. Or getting non-traditional data. So this is a paper that came out in L&O Bulletin literally almost a year ago, a little over a year ago, and likewise benefited from a bunch of awesome co-authors. And kind of the lead into this story is that, like I was saying, there's more publicly available data than ever before. But in particular, there's more high resolution data than ever before. And these data are becoming more and more common. But just like that Ledoux paper, working with these data sets is becoming more and more complicated. And remote sensing is a great example of these types of data. Where here I have an image from Peckel et al. What they did was they created a global surface water data set where they used Landsat imagery to track changes in the world's global surface water. Now, up until, I actually don't know. When the data first came out, they were exclusively hosted within Google Earth Engine, meaning that anyone who wanted to access these data needed to know how to code in Google Earth Engine, which there wasn't a lot of documentation at the time. And even if you were able to detect a trend, right, in change in surface water, ultimately you would want to contextualize that trend somehow. So you would have to merge those data with other climate or human population or dam data. And that could be really challenging, especially for folks that don't know how to code in Google Earth Engine or don't have access to high performance computing infrastructures. So what I had the pleasure of co-leading was the creation of the Global Lake Area Climate and Population Data Set, or GLCP, which basically does that, but for lakes. I'm going to walk through briefly what I mean when I talk about our data pipeline for creating this data set. But if you are interested in the nitty-gritty details, you can scan the QR code to access the scientific data paper. But in essence, what we do is we take hydro lakes, which is a compendium of over 1.42 million lake shapefiles, and we use those as a cookie cutter to extract lake surface area from the JRC Global Surface Water Dataset for every year, for every lake from 1995 through 2015. And we do that in Earth Engine, then we export it out into R for some local pre-processing or post-processing. We do a similar procedure using NASA's MERA-2 climate reanalysis data for temperature and precipitation. And we pair that with hydro basins, which is the basin equivalent to hydro lakes. And we use that as a cookie cutter to extract the basin's temperature and precipitation for every year from 1995 to 2015. And we do the same thing for human population with NASA's gridded population of the world data set. And then finally, all these outputs come together in one final join script to create the unified global lake area climate and population data set. Now, that's the down and dirty version, but the main takeaway from this is that we developed this workflow. We spent time manually and through manual and automatic investigation of QCing the data set so that other folks don't have to. The data are publicly available in a tabular format, accessible to linologists. You can download it right now from the Environmental Data Initiative. And while doing that, we tried to adhere to at least a spirit of fair data principles. Like I said, the data are publicly available on EDI, no paywalls, no restrictions. We retain unique identifiers in the data set so that you can join the GLCP with hydro lakes and hydro basins and hydro atlas literally with one line of R code. And as a result, all of our Google Earth Engine, all of our R scripts are publicly available. So if you want to add another variable from Mera 2 or something like that, you have all the scripts at your fingertips. And so we envision that the data set is highly reusable. There's going to be a version 2 of the data set coming down the line. It's at the monthly scale. We have a bunch more variables we're adding. I'll be talking about that at AGU if you're going. But I envision that the data set and data sets like this can be really useful for natural resource managers, folks that might not have thought about using these types of data. And one of those applications, and by the way, this is from like local to national scales when I'm talking about natural resources. So take, for example, one of my good buddies and a co-author on this paper did his dissertation at this lake. It's called Buffalo Lake. It's a large, deep oligotrophic lake in Washington State. And my buddy, he was interested, his whole dissertation was creating fish population stock estimates for this lake. Now, the thing is, in these types of lakes, the nearshore habitat is really important for things like breeding and energetics model for bioenergetics, predation. And so how much littoral area you have is really important for getting your fish population stock estimates. Well, managers like my buddy might be interested in understanding, number one, how Buffalo Lake compares to other lakes within the state. And also they might be interested in using the GLCP's time series to merge with co-located biological data to maybe explain how deviations in their population stock models corroborate with changes in lake surface area. And so we took a page out of Amina Pollard's playbook at the US EPA National Lake Assessments. And what you can do is you can create a distribution of lakes that occur within Washington State, where that's represented by this histogram. And the dotted line represents Buffalo Lake surface area. And now managers can look at this and say, okay, Buffalo Lake is representative of larger lakes within Washington State. And they can create a list of lakes that are at least comparable with respect to surface area. Maybe those might be more accurate, or more appropriate comparisons for Buffalo Lake. Additionally, they can create a time series of Buffalo Lake surface area in terms of seasonal, permanent, and total surface water. And once these are extracted, they can merge it with their own co-located biological data to explicate some of these trends. Zooming out to the national scale, this is one of my favorite figures from this data set. I believe that the data set can be useful for folks who are developing policies and drinking water policies, both at the regional as well as the national scales. Where what we have here is we extracted all of the lakes that occur within India. And I made a regression line through their change in surface area through the lake's time series. And there's about 11,000 lakes plotted on this map. And the dots are colored by the slope of change. What we see here is that lakes in eastern and southern India tend to be associated with increasing surface areas, and lakes in, I'll say, central, south central, southwestern India tend to be associated with decreasing lake surface areas. Now, if you're developing policies for drinking water or water allocations, on the regional scale, you might find this information useful because you can look around at your landscape and know whether your landscape is in an area where decreasing or increasing lake water area or lake water availability. Also, if you're developing policies at the national scale, you can take advantage of the GLCP being interoperable with its parent data sets and classify lakes by physiographic or even socioeconomic categories. So then you can develop policies around types of lakes that are not necessarily, that are maybe a little bit more flexible than geographic or geopolitical locations of lakes. And because we know that working with these data sets can be really cumbersome, one thing that we did was we tried to put all of this in a user-focused GitHub repo. So with this publication, there's one example that I skipped over that I can come back to if we have time. But all of the scripts that were used to make these sort of natural resource focused applications are publicly available in a GitHub repository. And we envisioned that this repo could be useful for folks that are learning to work with larger data sets, or even just empowering folks that never thought about working with larger data sets with at least a tractable hands-on example. Wow, I'm booking it. Okay, sorry folks that I'm going so fast. Okay, so switching gears a lot. Every example that I've talked about up until now, I've classified in my mind as being a little bit more computationally intensive. You know, we had that R script that iterated through 7,000 abstracts. We had this kind of complicated GLCP workflow. You know, it's 5 GB CSV. But ultimately, I view all of these techniques as something that's useful, all of these techniques as something that anybody can participate in, regardless of your data set size. And ultimately, it's the folks that are working in the field that they're how we get a lot of our data, that we do these meta-analyses or environmental syntheses. And so one thing that this paper came out about 2 weeks ago now, so we created a unified data set of co-located sewage pollution, periphyton, macroinvertebrate communities, even food web structures for Lake Valcon. And this is a data paper that came out of my dissertation. But the idea is how do we take field scale data or in situ data that tend to be really messy and really complicated, and make those data as hyper flexible as we can, so that folks doing similar research can still repurpose those data, and other folks can maybe use those data in a completely different way than we were ever intending. But the idea is, how do we take those inherently messy field data sets and make them elegant and flexible? And so, okay, this is when I get to talk about Lake Valcon. Lake Valcon, it's the lake of superlatives. It's ultra oligotrophic or naturally nutrient poor. However, in the past 10 years or so, there's been evidence of sewage pollution, largely centered in the southwest portion of the lake. But that sewage pollution is associated with small clustered human settlements. And a lot of really great work from folks like Olek Timoshkin, Nina Bondarenko, Tatiana Yakovleva have done a really great job of documenting these processes, but they've all been kind of anecdotal or based on more circumstantial evidence, even though they've done an amazing Herculean amount of work. But the thing is that it's Valcon, it's complicated. There's a lot of nuances that might occur, nuances in the biological responses that might be going on. But the awesome thing about Lake Valcon is that it's arguably the most biodiverse lake in the world, with high endemism, or critters that are found only at that lake. So for example, this is the slide. The world's only, oh no, a little thing is covering up its little eyes. There you go. The world's only exclusively freshwater seal. 11 endemic species of sponge, over 25 endemic sculpin species, and over 355 endemic species of amphipod. And I've worked with these amphipods for, oh God, almost 10 years now. But the thing is, it's this lake that just has incredible endemism, or exceptional endemism. It's very credible. But the thing is that these data and data about these organisms and how they're distributed and how abundant they are, especially in community compositional metrics, is not really well available, especially in a machine readable format. And I think the story that really hits this home when we were, wondering about writing this data paper, we're like, is this data set really unique enough? And then or uncommon enough. And I remember when I was at Griffith State University and I wanted these data, I was sitting in the bio department, like paging through like dissertations from the 1920s and like just writing these down, these data down, and like trying to create a makeshift data set that I wish I had in 2015. And so that's how we did. That's kind of what we did. I'm going to walk through what we sampled, but yeah, I'll walk through what we sampled. So here at Lake Baikal's southwestern shoreline, it's about a 40-kilometer transect, and those three main developments down there include the town of Istyanka with about 2,000 permanent residents, the village of Bolshoye, which has about 80 permanent residents, and the town of Bolshoye Galuslaya with about 600 permanent residents. And along this transect, I know this is a little small, that's not, there's about 17, there are 17 sampling locations. And there's the map on the next slide will show that a little bit more evidently. But it's at each of these sampling locations, or the nearshore sampling locations, where we wanted to get an idea of the various sewage metrics at each of these sites. So things like, I'm not going to go into details of IDW population, but suffice it to say the number of people that live at each of these settlements. At each of these sampling locations, we collected water column nutrient concentrations, so things like nitrogen and phosphorus. We sampled for PPCPs, so those pharmaceuticals and personal care products. Note that are very rare, that very few lake studies actually sample for. And also things like stable isotopes, so things like N15, which tends to be associated with sewage pollution. We also, it's not included in this, but also microplastics within the water column we also sampled for. And then to get an idea of how biota might be responding to at each of those sites, especially with our sewage gradient, we tried to get metrics of community abundance. So things like the periphyton algal community composition, macroinvertebrate community compositions, all those nice little amphipods that I showed you on the previous slide. But also recognizing that changes in community composition can ultimately lead to changes in how community members interact with one another. We also tried to get an idea of how the food web might be shifting with increasing sewage pollution. So we have stable isotopes for trying to get an idea of those food webs, stable isotopes of the periphyton and the amphipods. But we also have fatty acids. There's about 55 different fatty acids that we sampled for, both within the periphyton as well as within the grazing macroinvertebrates. And you can ultimately use those measurements to get you to build food webs within the littoral zone, who's eating who. But okay, so that's kind of boring, talking about like all the things we sampled for. The main takeaway is that all together, we have about 150 different variables at each of these sites. And actually, even though that we had an analysis in mind when we collected all of these data, we still collected them. And we know that those data could still be repurposed for other people. And even more studies that we have in mind down the road. And so one of the things that I think that those data could be repurposed for, so see, these are the sampling locations. They could be used to fill gaps within current monitoring efforts that are going on at Lake Baikal. So our good colleagues, Alek Timoshkin and his group, they sample much of Lake Baikal's 2,100 kilometer shoreline for periphyton community compositions. And I think it's data sets like ours that can be pretty flexible, could be used to fill gaps where if they weren't able to sample our locations at our sampling times, we could help fill in their current monitoring efforts, or exercises like these could be used to help current monitoring efforts. Additionally, we also included some food web metrics, things that current monitoring efforts don't include in their samplings. And so they could use these data to ultimately spur new sampling collections for planning for future field seasons. But I think the main thing that I take away that I wanted to contribute is kind of this data product ecosystem. Where for this data product, we have an open science framework project. We have a published EDI portal or repository, and we also have an archived GitHub repository as well as the data paper. And each of those pieces kind of plays a role in this data product. We're on the OSF or Open Science Framework portal. That contains all raw data like you would find in the field. All the misspellings, all of the poorly formatted Excel spreadsheets, as well as pictures of our field sampling locations and pictures of our field notebooks from every day in the field. So that if you want to go back and look at exactly what we did from the raw data in the field, misspellings at all, you can. However, the clean, analytically ready format live on EDI, where you can find all of our data at the replicate level. They're spread across 11 different CSVs. However, if you are new to merging those different CSVs together, the GitHub repository contains 8 different analytics scripts that we use in our analyses, but they can serve as templates for how you can combine these data sets in different ways to get at your specific research question. That was about 7 years of work right now. Okay, so, and by the way, just one quick plug, since we have a little bit more time. The whole reason this data set even came into existence was because of my Russian language skills and all of the training. So like, for folks that are often like, oh, wow, I don't have to take Spanish or something. All of the taxonomic keys, all of the resources for identifying these taxa, the fatty acids even, were all in Russian. So like that all had to be translated. And I was the only Russian speaking person that was leading this project. So kind of cool to have this come about. Okay, so that's the end of my researchy talk, and then I promised that I would also offer resources. And so trace resources that are specific to Washington State University, but now that everything's online, anybody can access it, right? So one of the things that I'm most proud of is the creation of this R working group. And what that is, it's kind of a grassroots organization of faculty, staff, grad students. It has about 200 members, but every week, folks get together Wednesdays at lunch, and they discuss either a package, a research profile, which is like, hey, look at some cool things I did with my research in R, or troubleshooting, which is like everybody comes together, they say, I have an error, and we crowdsource that error. Or even like, hey, I have some data, I don't know how to analyze it and graph it or plot it in a way that makes sense. And then we help that person come up with an analysis or build a plot. You can join the listserv. They're all on Zoom. All the meetings are on Zoom now. And the schedule changes from semester to semester based off of what the community wants. And we've had everything from what I call fundamentals. So things like flipping tables, pivoting tables, all the way up to multivariate time series analysis. And it's purely based off of what the community wants. And it's always the most awkward meeting. But the first meeting is everybody writes their interests. And then you stand there and say, okay, who can give a talk in this? And then you stare at people until somebody volunteers. But it's been really successful. And then also we have a Slack workspace, which functions, I assume you're studying at University of Wisconsin, functions similar to like a math center or a writing center. But we kind of envisioned that this would be like a data center, where if you have questions about how to aggregate your data, how to publish your data, you can toss it to the Slack workspace, and we can help you figure it out as a group. And there's currently about 90 members in the Slack workspace, I think. So yeah, I'm no longer the lead of the R working group, obviously, now that I'm here, but the 2 people who are in charge of it now are really good folks. So I highly encourage that if you folks are interested. There's also an analogous Python working group that does similar things at Washington State University. We also, I was one of the folks that helped create a reproducible science workshop. If you join the Slack workspace, there's it's 24 hours of training material that walks through everything from tidyverse in R to how to use GitHub. I made a module on linear and nonlinear modeling. And then there's also a module on targets which is a dependency tracking software. I can talk more about that later, but basically it's just like how you can create an ecosystem of scripts where like if your computer knows which data products have changed between runs. So it knows, okay, don't run that script because everything's the same. Run this script because something changed. So we make sure that everyone is at least, they've at least seen it before, right? It's not how smart you are, it's just the number of times you've seen it. We also conducted HPC trainings at Washington State University, where we could usually get most folks up and running on the HPC clusters within 3 hours. And then also we started a special topics workshop for Google Earth Engine. Robert gave a process-based lake modeling seminar. Yeah, so hopefully that stuff continues. But the main takeaway is these are all resources and training. It's open to everyone, even though I say it's at Washington State University. And there's a lot, like last I checked here, the Slack workspace has over 80 hours of recording material for folks interested. And then also something that really inspires a lot of hope for me is the advent of virtual conferences and virtual summits. And you know, this has gradually happened over the past year, 20 months. Places like Ecological Forecasting Initiative, Knowledge Guided Machine Learning Workshop, NEON, and GLEON, the Global Lake Ecological Observatory Network, have done a really great job of transitioning from in-person conferences to a virtual workshop or virtual conferences that ultimately allow for more engagement and more participation. Or, you know, at the very least, it takes a lot of the hassle away, especially when you think about the travel fees and lodging and visa requirements for a lot of folks, especially for an international organization. But something I want to talk about, I think is pretty cool. I think this has also created a space that has allowed other new workshops and conferences to come into existence. And I'm really inspired by all this awesome stuff that I'm seeing. And one that I've had the privilege of being involved in was the creation of the Virtual Summit for Incorporating Data Science and Open Science in Aquatic Research, DSOS for short. And the way this came out was my good buddy Jake Zwart, who is a data scientist at USGS. He and I just kind of saw all these aquatic science conferences being canceled or postponed. And we're both early career people who really wanted to share our work. So we said, why don't we try to transition from that in-person format to a virtual format? And we figured like 40 people would show up, probably nothing big, but actually in 2020, we had over 425 people register with about 150 people attending the virtual summit. And that was in 2020, and that was just me and Jake doing that together. And we said, you know, we think we found, oh, by the way we did it, how we did it, down here at the bottom, we have an L&O bulletin piece that came out a year ago that basically details what we did, what we think worked well, what we thought could change, and maybe some plans for the future. But speaking of the future, for the 2020 summit, we got some more folks involved because we think that we really tapped into something special here. And so it included Robert Lavig, postdoc at University of Wisconsin-Madison, Carol Barbosa, who's a PhD candidate at University of Sao Paulo in Brazil, Bella Olexen, who's a postdoc at University of Wyoming, and Allie Kramer, who is now a postdoc at University of Washington. And we wanted to create a 2021 virtual summit. But the thing is, one of the feedback we got from one of the main pieces of feedback that we got from the 2020 summit was that this is all great, but how do I actually do the techniques that you're talking about and the data science techniques and open science techniques? And so we teamed up with the AMON-J, or Aquatic Ecosystem Modeling Junior group that included Patricia Tran, who's a PhD candidate here at University of Wisconsin-Madison, Jorrit Messman, who just finished his PhD at University of Geneva, and Johannes Feldbauer, who is a PhD candidate at Technical University Dresden. And we created this combined DSOS AMON-J Hacking Limnology workshop and the virtual summit, where the Hacking Limnology workshop was the week before the summit and was 4 days that had hands-on coding exercises as well as keynote speakers. in remote sensing, big data that was hosted by Neon, machine learning, and numerical modeling. And each day had a basic fundamental intro talk so that folks could get used to the vocabulary and the ideas. It had a keynote talk that talked about like, these are the things that you can do with these techniques, but most of the time was spent on hands-on coding exercises, as also some like brainstorming and discussion sessions to get new ideas generated among the community. And this, like the previous year, also attracted a large audience globally. So this is a cartogram where countries are colored and sized by number of registrants. We had both 686 registrants, and each, I think there was between like 75 to 100-ish people per day. And we actually, I think this came out a week or two ago, had an Elena Bulletin piece coming out that documented similarly what happened, what worked well, what didn't work well, and what to do for the future. But the main takeaway here is that I think that this is a great example of, given everything, of everything that's going on, the disconnection from how isolated a lot of us are, we can still remain connected through this type of virtual science. So science can still keep going and it ultimately lowers a lot of barriers for other folks. One of the things that was really great was, about 60% of registrants were outside of the United States. Outside the main countries outside of the United States were Nigeria with about 20% of registrants. Germany, I think, was about 5%. Canada was about 6%, and Brazil was about 6% as well. So I don't really have a big conclusion, grandiose conclusion slide, but I think the main takeaway is that going back to that introduction, right? Like 2020 really sucked. And 2021, it's getting better, but it's still not where it could be. And there's a lot of things that we could think negatively about, but at least like with this type of techniques, and at least we live in an era when there's so much publicly available data than ever before. We can spend this time investigating already collected data sets and already existing data sets to get at new skills, and especially thinking about systemic inequalities. And like when I think about what is the good that I can produce in the world with my science, I'm not saying that this is a solution to anything, but I'm just saying it's what Michael Meyer right now with his science can do. I know that for a lot of groups, it can be dangerous to go out in the field, like for Black, Indigenous people of color, LGBTQ+ scientists. And so, this might present a way that a lot of folks, especially folks that might have various physical challenges, that they can engage in the environmental sciences in a really energetic way. So we have the potential to increase inclusivity through the science as well. So I'm going to stop rambling, but thank you.